Great. Thank you, everyone, for being here. And I want to thank the organizers of the Women's SIG for inviting me to present today on the research that's ongoing in my lab. Before I begin, I want to just say I have no disclosures, sadly. And I also want to acknowledge my trainees, staff, collaborators, and funders who contributed to the research I'm going to talk to you about. So a bit about me. So I'm a professor at Toronto Metropolitan University and a CRC Tier 1 in sex, gender, and diversity in brain health, memory, and aging. And I got into the research on sex, gender, and diversity by accident in some ways. So I've been, since my PhD years, been doing research in the cognitive neuroscience of memory and aging primarily. And in the 2010s, I noticed that most of the research in the cognitive neuroscience of episodic memory and aging focused on comparing extreme age groups, so looking at differences in brain activation and behavior in young compared to older adults. And there was very little known at that time, at least, about how midlife was affecting brain and cognitive aging. And so with that in mind, in the early 2010s, I conducted a study called the Montreal Adult Lifespan Study in collaboration with many others, and it was led by my former PhD student, Lizzie Ankudovic, where we had 131 healthy adults between the ages of 19 and 79 come into the lab into a task fMRI study where they performed different types of episodic memory tasks. So episodic memory is our ability to encode, store, and retrieve past personal experiences in rich contextual detail. So for example, remembering where you saw your friend last, so the association between a person and the location, so that I refer to as spatial context memory. The order in which you might have bumped into colleagues here at OHBM, and so that I would refer to as temporal context memory. So my lab has really been interested in understanding what are the neural bases of spatial and temporal context memory and how does it change across the Montreal adult lifespan. So we had these adults perform a spatial context memory task and a temporal context memory task that differed in difficulty to account for performance effects and their interactions with age. And I'm not going to talk to you about the imaging results. You could refer to the papers here by Lizzie for the imaging results, but I'm going to give you the behavioral results because they framed the main study that I'm going to talk to you about today. So when we looked at the performance of adults that are young, so this is 18 to 39 years of age, middle-aged, 40 to 60 years of age, and older, 60 to 79 years of age, we noticed that, yes, there's somewhat of a linear decrease in spatial context memory, but when we looked at between-group comparisons, what really surprised me at that time was how poorly the middle-aged adults were doing. So middle-aged adults were doing significantly worse than young adults, but not performing significantly differently from older adults. So this really highlighted how midlife is an important period in our adult development, at which memory decline first emerges, and that's really important within the context of dementias and Alzheimer's disease particularly. So many of you might have seen this figure or are familiar with this figure, and it really is the argument that Alzheimer's disease, the neuropathology associated with Alzheimer's disease, and probably other dementias as well, emerges many decades before the initial symptom onset, and how midlife is probably the critical period in which risk factors, either modifiable or non-modifiable, might change the direct trajectory in which people travel across their adult lifespan. And the other important thing to consider is at midlife, the risk factors for Alzheimer's or developing Alzheimer's is twofold in females. And in fact, 2/3 of cases in Alzheimer's disease is diagnosed in females. So when you put these things together, midlife being a critical period in the aging trajectory, and there being a sensitivity to the development of Alzheimer's disease in females, starting at midlife, you of course have to think of menopause, something that all females with ovaries experience traditionally in their midlife, around the age of 50 to 52. So what is menopause? I know we talk about this word, but very few people understand what that is. So menopause is defined clinically as the cessation of your menstrual cycling for 12 months or more. But there's a period, perhaps even a decade-long period prior to that, where there's much fluctuation in your sex hormones, particularly estrogen, progesterone, and the follicle-stimulating hormone, that affects not only your brain, but your whole body. So these are ovarian hormones. It affects your heart health, it affects your kidney health, it affects your whole body, and it affects your brain. And so we wanted to understand, how does menopause or reproductive aging in females interact with the effects of chronological aging to influence brain health and cognition and midlife? And so with that in mind, we designed the Brain Health at Midlife and Menopause study. This is an effort that involves a lot of collaborators. So I'm the PI, but there's many collaborators from different countries on this grant. And see, this is, yeah. Sorry, there was like a hiccup in the past on this slide too. Can I go backwards? Okay. Yeah, all right, thank you. And so the slide that kind of got blurred there is that the main purpose of this study is really to explore the question of whether menopause can be a window for identifying females that are at greater risk of developing Alzheimer's disease later in life. And so this study started as a cross-sectional study, but now we are trying to expand it into longitudinal. So we have one cohort that is being followed up, and we're looking for additional funding to expand the longitudinal component. But to answer that big question, we actually have to answer many multiple small questions. So first, we need to show objectively, how does biological sex and menopause status affect episodic memory? I love episodic memory. There's many other cognitive domains we could look at as well, but in my lab, we look at episodic memory. And then how does biological sex and menopause status then affect the neural correlates of episodic memory at midlife? Do any of the observed effects interact with either modifiable risk factors such as vascular health, inflammation, sleep disruptions, all these things that are modifiable in your midlife? And does it interact with non-modifiable factors such as your genetics, such as having an ApoE gene, ApoE4 allele that puts you at increased risk for Alzheimer's disease? And then the final question, which is like long haul in a decade, hopefully you'll invite me back to answer this question, is to really understand from the precision medicine perspective how individual differences in lifestyle factors and social and structural determinants of health interact with all this to really help us understand how an individual with intersectional identities experiences menopause and midlife, and how that might relate to differential patterns of aging. So this really speaks to a second passion of mine. I'm interested in sex, gender, and diversity. So as we all know, a lot of the cognitive neuroscience of aging studies have not been done in a very inclusive manner. And so the BAM study is really focused on integrating individuals from different cultures who live in Canada. So in Toronto, there's a large population of South Asian, Caribbeans, East Asians, like that are part of our communities in Canada, and they are not being addressed by the current models of aging and dementia. So I'm really passionate about making sure our research reflects what's going on in different communities as well. So it's a long haul. Today I'm going to talk to you about results from the first 3 questions. The 4th question, like I said, give me 10 years and maybe we'll have the sample size to do that. So a bit about BAM. So what have we done so far? So BAM has been funded since 2017 by the Canadian Institutes of Health Research and the Canada Research Chairs Program. We've enrolled 2,460 adults into this project so far. From that number, we've had 546 adults come in for a behavioral session where they do many questionnaires. I'll give you a bit more details in the next slide. And then they also undergo multimodal MRI acquisitions in session 2. So there's 298 adults for which we have that data. So in session 1, which we have 546 so far, we have blood draws. So we do use this to do hormonal assays, to look at like several types of proteomics, metabolomics, and also blood-based biomarkers for Alzheimer's disease. We do neuropsychological and psychiatric testing to ensure that individuals are not underdiagnosed for these disorders. What's unique, I think, about BAM is that we really came into it with reproductive health and women's health in the forefront of our mind. So we do a lot of questionnaires to understand the reproductive histories of our females, when they entered menarche, how many children they had, whether they have a history of PMDD, and then where in the menopausal transition stage are they. And we use the STRAW 10 criteria to define that behaviorally and clinically. but we back it up with the hormonal assays to say, yes, they are, premenopausal, perimenopausal, or postmenopausal. And so that's the data from session 1. And then for session 2, session 2s in these kind of larger programs, they're going to happen 3 months later, 4 months later. And this is a very sensitive transition in a female's life. And you can be perimenopausal and also postmenopausal when you come in for scanning. And so we do the reproductive questionnaire again. We do a health follow-up again. And then we have individuals do a series of MRIs. So we do structural, so the typical MP ranges. We do ultra-high T2 scans to look at hippocampal subfields. We do resting state bold fMRI for 10 minutes, eyes open, with all the breathing and everything recorded at the same time. We do task-based bold fMRI. This is for episodic memory, which, I will die doing episodic memory research, right? So task-based bold fMRI for episodic memory, and then diffusion MRI. And then we have debriefing questionnaires to understand how the person experienced their MRI task, and then whether they were stressed, whether they mind wandered, all of that. And then we do the blood draw at the end of this session. So what's unique here is that we have time-locked data. So this is not like other big databases where the genotyping, the biomarkers, the hormones are asynchronous to the MRI acquisitions. This is time-locked to the MRI acquisitions, and the behavior, the larger data sets, are within 3 months of the data collection of MRI. So where are we at so far? So of the 298 that have been scanned, we have screened 270 of them so far. And today I'm going to talk to you primarily about the middle-aged males, middle-aged females premenopausal, and the middle-aged females postmenopausal. The perimenopausal we're analyzing right now, they really do not fit in a linear model. They have to be analyzed separately from pre- and postmenopausal. There's a lot of variability at perimenopause. So what's the task fMRI paradigm? These are middle-aged adults. We can make it a bit tougher on them in the scanner, right? And so we wanted to do, again, I love spatial context memory, so it's a spatial context memory task still, but it's way more challenging. So we have to encode a face in one of the 4 quadrants. You're told this, memorize the face and its spatial location in one of these 4 quadrants. Also, simultaneously, to make sure they're awake in the scanner, tell us if you think this face is pleasant or neutral. They have 2 levels of difficulty, encoding either 6 or 12 faces. Then there's a break where they give a subjective assessment of what they think their memory was, and there's a break of 1 minute. And then we do the retrieval run. So this is like a hybrid working memory, episodic memory paradigm in some ways. And during retrieval, they see one face in the center of the screen. It could be an old face or a new face. And they're asked to tell us whether they remember the face and its spatial location. Yes, it's on the top left, top right, bottom right. Or they know the face, but it's just familiar. They don't remember the location of the face, or the face is new. And so what's interesting about this behavioral paradigm is that it allows us to differentiate different types of episodic memory. So we could look at recollection, the ability to look at face space locations, and this is important. It actually tracks to different parts of the medial temporal lobes, structural and functional integrity. You could look at recognition, so the failure to remember spatial context information, but just remember seeing the face. And then you could also look at novelty detection, the inability to differentiate new from old. So we look at all these different behavioral paradigms, but I'm going to focus on context memory in this talk and on recognition memory. So the first question we had was, are there sex differences in midlife? Because behaviorally, in the older adult samples that we've looked at, there really haven't been historically sex differences. But when you zoom in on midlife and you enrich your sample for females at different stages of menopausal status, you really do see a difference in the effect of age on memory-related brain function in males versus females. So this is work done by postdoc Ricky Lissaman, showing that using linear mixed effects modeling, that females have a greater effect of age during midlife. Now, is this related to menopause? So using segmented regression, postdoc Annelise Laplume basically didn't code for menopause, but she looked at within the females and males' aging trajectories, is there an inflection point in which memory decline emerges? And in females only was there an inflection point for source memory, and it occurs around the age of 50, roughly the average age of menopause. And so when you do kind of project our individuals based on menopausal staging, you see that that age effect really is being driven by females at at postmenopause. So the way we're thinking about this is that the effect of menopause or reproductive aging on chronological aging is really to kind of act like lighter fluid and accelerate the effect of chronological aging in females at postmenopause. So the next question is, what's the neural correlates of this? So this was a project run by a master's student, Ariel Crestall, in which we explored how does menopause status and age within group affect memory-related brain function. So to do this, we used a behavioral partial least squares analysis, which is a multivariate method, where we put two behavioral vectors, the vector of age and the vector of performance. So here we have at the second graph, spatial context accuracy, and then at the bottom graph, recognition accuracy. So these are the flip side of each other on this task. So if one goes up, the other usually goes down. And we identified a network of brain regions that really was related to memory performance in postmenopausal females uniquely. So increased activation in these blue areas of the brain, so ventral occipital cortex, medial temporal lobe cortex, activation of these areas support context memory. But with increased age at postmenopause, activation in these areas decrease. And so this is kind of modeling that interaction that I showed you where the effect of age is accelerated at postmenopause. So why are postmenopausal females doing poorly on our task? Well, the age-related effects on medial temporal lobe and ventral visual occipital cortex seems to be increasing at postmenopause. Now, the next question is, well, how does this relate to modifiable and non-modifiable risk factors? So this is work done by PhD student Julia Curley, where she looks at vascular risk factors. So anyone, I'm doing me search at this point, so anyone going through menopause or who has gone through menopause knows that there's a lot of vascular effects in menopause. So you have increased adiposity, you have poorer cardiac function, there's also an increased risk of obesity, higher BMI. There's a lot of vascular risk that occurs at menopause transition. So what we wanted to know is, and I want to take a break here to say menopause is not a disease, right? And there's a lot of variability in menopause and how females experience menopause. So if we're looking for females that are going to convert to Alzheimer's disease, obviously not all females do, right? So what are the factors that relate to the poorest functioning individuals on our episodic memory task? And so looking at that, we looked at vascular risk factors, and what we saw, this is work that's been published, is individuals that had a high CADE score, so this is vascular risk that predicts dementia at midlife, were more prevalent in the postmenopausal age. So yes, you have higher vascular risk at postmenopause. But then when we did a linear mixed effects modeling with Helmert contracts so we could compare different stages of CADE score, what we saw is that females with CADE scores of 8 to 9 and above, so really high vascular risk, are the ones that are really driving those memory effects that we're seeing at postmenopause. So it's not just menopause, it's how healthy you are, how healthy your heart and your vasculature between your heart and brain kind of connections is at menopause. So I'm going to wrap up now. So what I hope I've kind of communicated to you is menopause is important. It accelerates the effects of age and alters brain function and episodic memory. And it is the effects of menopause on cognition are related to vascular health. But more generally, what I hope I have communicated to you is how critical it is that we look at biological sex and sex-specific experiences in life, such as menopause, such as pregnancy, such as, retirement, like we saw in the previous, like these sex-specific experiences that might happen that have differential effects. So how retirement have a more profound effect in males compared to females, but perhaps these sex-specific biological transitions have a greater effect on females. Well, clearly because they're female-specific. Now, when developing our models of brain aging and dementia, we have to consider these things, and we also have to consider diversity in these experiences across communities. And that for advancing precision medicine, you can't do precision medicine without doing women's brain health research. So I hope that motivates all of you to look at this kind of work in your projects as well. Thank you.